All right, today we're going full-on diagnostic detective. We're on a mission to unmask the microscopic culprits that cause disease, and we're going to follow the clues all the way from the patient sample to the final critical ID. So, let's grab our magnifying glass and get this investigation started.
It really all boils down to this one question, doesn't it? I mean, when your prime suspect is thousands of times smaller than the head of a pin, you can't just slap on the cuffs. The whole investigation needs incredible precision, a solid strategy, and a bunch of clever tests to basically force this invisible killer to show its hand.
Every good investigation starts at the scene of the crime. Well, in clinical microbiology, that scene is the patient sample, the specimen that shows up at the lab. This right here is our ground zero. And just like in a real case, the entire investigation lives or dies on the quality of that initial evidence. I mean, look at these examples. A leaky container, a mismatched label. That's like contaminated evidence at a crime scene. It's going to send the detectives down a completely wrong path, wasting precious time and, potentially costing a life. So, to protect the integrity of the investigation, labs follow what we can call the Goldman rules. First up, the ID has to be perfect. Labels have to match forms, no exceptions. Second, time is everything, which is where that 2-hour rule comes in. And finally, quality is non-negotiable. It's all about preventing, a diagnostic miscarriage of justice. And of course, let's not forget these microbial suspects can be really dangerous. So the number one rule for these lab detectives is don't become a victim yourself. That's why they follow what are called universal precautions. Basically, treat every single sample like it's infectious. And they work inside these biosafety cabinets, which are like high-tech containment fields. A typical clinical lab uses a Class 2 cabinet, the one you see in the middle there, which protects the person, the environment, and the sample. Pretty cool.
Okay, the evidence is secure, we're all suited up and safe. Now it's time to find our first real clue. For more than a century, the single most important first step has been a technique called the Gram stain. This is the pivotal test. It's what splits the entire bacterial world into two massive rival gangs.
Think of the process itself as a kind of chemical interrogation. First, you hit all the bacteria with a purple dye. Then, you add iodine to lock that color in tight. Now, step 3, that's the critical one. You use alcohol to try and wash that color out. And finally, you add a pink counterstain to color any of the bacteria that lost that initial purple dye.
And here's the brilliant result of that interrogation. Bacteria with a thick cell wall, we call them gram-positives, they hold on to that first purple dye like their life depends on it. But the bacteria with a thin wall, the gram-negatives, they can't hold it. The color gets washed right out and they end up picking that final pink stain. And just like that, we know which family our culprit belongs to. Are we on Team Purple or Team Pink?
All right, our first clue is in. The culprit is purple. So we're officially on the trail of the gram-positive family. But that's still a huge group of suspects. So now it's time to start profiling them to really narrow this thing down.
And this decision tree you're looking at, this is the detective's roadmap. For our purple round suspects, also known as Cocci, the very first question we ask is the catalase test. We add a chemical. Does it bubble? If it does, that's a positive result, and we're dealing with the Staphylococcus gang. No bubbles? Well, then our suspect belongs to the Streptococcus family.
So let's say we follow that bubble trail. It often leads us to a most wanted suspect, Staphylococcus aureus. So how do we confirm its identity? We run 2 more tests. First, the coagulase test. We check if it can clot blood plasma. Yep, it can. Then, we grow it on this special plate, or agar, that has a sugar in it called mannitol. Well, S. aureus loves to eat this stuff, and when it does, it turns the plate this amazing golden yellow color. That's literally how it got its nickname, the golden pathogen. Case closed on that one. But hold on, what if our first test was negative? No bubbles. Then we're on the trail of the Streptococci family. And for this branch of suspects, our next big clue comes from watching how they behave when we grow them on a plate of blood agar.
This behavior is called hemolysis, which is just a fancy word for breaking down blood cells. It's a key part of their method of operation, or MO. You've got beta hemolysis, which is total destruction. It leaves a clear zone. Then there's alpha hemolysis, which is just partial destruction, creating this kind of greenish look. And finally, gamma, which means no hemolysis at all. Each one of these patterns points us to a different group of suspects.
So far, we've only been profiling one type of suspect, the purple round ones. But believe me, the microbial underworld is vast.
So let's quickly flip through the case files on a few other notorious culprits, just to see the different kinds of clues they leave behind.
Okay, so some suspects are rod-shaped and they have this superpower. They form these incredibly tough protective spores that let them survive almost anywhere. A really infamous example is Bacillus anthracis. Yep, the bug that causes anthrax. And when you grow it in the lab, its visual signature is just unmistakable. You get this wild, wavy Medusa head colony on the plate. No mistaking that.
Then you've got other rod-shaped bacteria that don't have that superpower. So to identify them, we have to look for other behaviors. Take Listeria, for instance. It's identified by its unique tumbling motion when you look at it under a microscope. And I have to point out the clinical alert here because this is so important. This bacterium is a severe danger during pregnancy.
So what about that other gang, the pink-staining gram-negatives? Well, they have their own interrogation methods too. For suspects like Neisseria, it all comes down to their diet. We basically offer them a buffet of different sugars and see what they eat. If the color changes to yellow, that means they're fermenting the sugar. As you can see, N-gonorrhea only goes for glucose, but its cousin N-meningitis eats both glucose and maltose. And that's exactly how we tell these two apart. So, through this whole systematic process, staining, testing, profiling, the detective just keeps narrowing down the possibilities one by one until only a single suspect remains. And then, the case is ready to be closed.
But look, that final report, it is so much more than just a name on a piece of paper. That identification is the crucial piece of intel that lets a doctor choose the right weapon, the right antibiotic. If you choose the wrong one, the infection could win. But if you choose the right one, you save the patient's life. It's that simple.
But the story doesn't always end there. Oh no. Sometimes identifying one of these microscopic culprits completely blows open a different case. It uncovers a much bigger, much more sinister secret that's been hiding inside the patient. Let me show you one final absolutely stunning example of this. Meet Streptococcus bovis. It's a member of that strep family, one that doesn't harm blood cells. But here is the truly incredible connection. If a lab detective finds this specific bacteria in a patient's blood, it sets off a massive alarm bell. Why? Because it has an incredibly strong link to colon cancer. The bacteria is basically the first informant, tipping doctors off that there might be a hidden, deadly tumor. It's just amazing.
So you see, it's never just a lab test. From a simple stain to a whole series of biochemical tests, every single step is part of a larger detective story. It's the process of finding that final definitive clue in what is truly a life-saving mystery.